So in this presentation, we want to look at enzyme regulation. What is very important about this topic here, or this subtopic, or this concept, is that you don't want enzymes to work endlessly without stopping. So what mechanisms do we have in the body that makes sure that there is a stop to enzymatic action? Okay, or if we can't say that, what do we have in the body that makes sure that the rate of enzymatic activity slows down or basically increases? So we have concepts to do with these mechanisms that we want to look at. So we start with this scenario. We are having an enzyme. This enzyme is actually very important in genetically modified maize. So what this enzyme does is basically regulating nitrogen metabolism. We know that nitrogen is actually very important in plants. So what this enzyme called glutamine synthase does is to regulate this metabolism, this nitrogen, in this genetically modified maize. So scientists have actually noted that this enzyme is actually very, very active when there is deficiency of nitrogen. What the scientists also discovered was that there are other small molecules in this maize that bind to this enzyme and change its shape, and then making its enzymatic activity faster. So enhances enzymatic activity. So in a separate experiment also, you have scientists also discovering that when you have glutamine, when you have it in large amounts, it basically reduces the activity of this very enzyme, glutamine synthase. Okay, so even when you have high substrate levels, so the presence of glutamine makes the enzyme not to work very well. Okay, but the enzyme works very well when nitrogen is actually deficient, in nitrogen deficient conditions. Okay, so the things that we have talked about actually rotate around issues to do with enhancement of enzymatic action or a decrease in enzymatic action. These are the things, and I think you can now categorize this. Now, where do we have this enzyme working very fast, and where do we have it being inactive? So you have those two things, or two conditions. So that means that we are talking about regulation of this enzyme. Under what conditions should we have it working efficiently, and under what conditions do we have it working less efficiently? And that's why we're talking about this regulation. So one of the regulation types that we shall talk about, and that we actually need to know in this presentation, is what we call allosteric regulation. So allosteric regulation is what we need to know and how it basically happens. And then the second one is covalent modification. Okay, covalent modification. What is it? We also need to understand that. Okay, the other thing that we really need to know is an issue to do with feedback mechanism. So when we talk about feedback mechanism in regulation of enzymes, what is it? Can we explain it? And why is it very important in maintaining balance? Then lastly, we need to address issues to do with post-translational enzyme regulation. What advantages do we have in this concept in relation to regulation of enzymatic action at the gene level? Because we shall also look at that gene level regulation of these enzymes. Okay, so that is basically it. Still, this question again points to real application of enzymes in at industrial level. Okay, and in this case, we are basically looking at the application of enzymes in getting what we term as GMOs, genetically modified organisms. And in this case, we're talking about genetically modified maize. So allosteric regulation, when you have a molecule that binds to the enzyme and then either making it to work efficiently or making it not to work efficiently. So we can have positive regulation or negative regulation. Okay, so we can have positive allosteric regulation or negative allosteric regulation. It's about binding of another molecule to the enzyme on a site that is other than the active site, and then altering the active site. If it alters the active site such that it can efficiently bind with the substrate, that is positive. If it binds to the other part of the enzyme and then resulting into a change that will make it not to be able to bind with the substrate, then that is negative regulation. Okay, so covalent modifications, again similar, but here the bonding between the enzyme and the other molecule is a covalent bonding. Okay, so again, it results into alteration in the enzyme shape and then eventually either rendering it inactive. So the other difference between allosteric regulation and covalent regulation is that for allosteric regulation, the interaction is not covalent and is reversible. Again, on the other side, actually, so I think you can be able to illustrate that using a diagram. Just draw the enzyme active site, and then you have another molecule that attaches to another part apart from the active site. And then in our example, glutamic synthetase, can you explain feedback mechanism using that example? It's just very, very straightforward. Accumulation of glutamine makes it impossible for, or makes it very difficult for the enzyme glutamic synthetase to basically work. Okay, so that is a negative feedback regulation that we are having in this case. And then the other question is describe the advantages of post-translational enzyme regulation over regulation at the level of the gene expression in relation to plants growing under nutrient stress. So what is post-translational enzyme regulation? Post-translational, the point is at translation. So when we say post-translational, we are meaning after translation. Okay, so after translation, remember translation is a part of the process of making proteins. So what happens in translation is that translation happens after transcription. After transcription, we have translation. So translation is when now amino acids are actually being assembled to make this protein. But as this assembly is taking place, before the final synthesis, you'll have mechanisms within the cell to cross-check to see if it is the actual amino acid or it's the actual protein that is actually being synthesized. If there are errors, there are some improvements. Some amino acids may actually be removed and things like that. Okay, so that is post-translational. That is what happens there. So of what advantage is it after over gene expression? So remember, gene expression is actually the first thing to happen, because you will need to have transcription happening. After transcription happening, then you have translation. So when we talk about gene expression, genes express themselves